Hi everybody. Today we are going to start learning about the cell and cell organelles. So let's go ahead and get started. Make sure you have your notes. So what we need to learn about the cells, first of all, is that there are different types of cells. So there are prokaryotes, which you see up in the upper right corner. Prokaryotes have no membrane-bound organelles. They're pretty simple. These are cells like bacteria. They have DNA sort of floating around the middle. They have a membrane. They've got cytoplasm and ribosomes, but they don't really have much else. We have an entire chapter devoted to prokaryotes later in the year. So what we're going to focus on mostly are eukaryotes. Eukaryotes have membrane-bound organelles. This is the big definition that you should remember, that there are membrane-bound organelles found in eukaryotic cells. There are two types of eukaryotic cells that we're going to focus on: animal cells and plant cells. So you can see here the eukaryotic cells are much more complicated than the prokaryotic cells. There's more things inside. There's more going on. And the big key thing here that you should be looking at is that the eukaryotic cells have compartments, right? There's a compartment here, there's a convoluted compartment there, this is a compartment, in the other cell there's compartments. So eukaryotic cells compartmentalize many of the chemical reactions that go on in the cell, and that helps make the cells more efficient. So animal cells, plant cells, you can kind of see how there must be a difference between the two that we'll talk about later. But they have compartments, membrane-bound organelles, which are basically just compartments. So from your reading, you should have learned about a few major differences between prokaryotic and eukaryotic cells, and you should be able to classify some of these as prokaryotic or eukaryotic. Let's take a moment to answer these two questions now. So our first question is going to ask about the major differences between pro and eukaryotic cells. The big difference you need to remember is that definition we talked about before. Eukaryotic cells have membrane-bound organelles and prokaryotic cells don't. In terms of these, are they prokaryotic or eukaryotic? A maple leaf is a plant cell, that's eukaryotic. Stomach cells are part of animals, that's eukaryotic. A bacteria is a prokaryote. Amoebas are single-celled eukaryotes, so they are eukaryotic, and mushroom fungi are eukaryotes. So really the only prokaryotic thing present is the bacteria. All right, in terms of the differences between eukaryotes and prokaryotes are much smaller, right? You can see there a bacterial cell is pretty small compared to an animal cell or even a plant cell. And we can see that they're at least 10 times bigger. So eukaryotic cells are quite a lot bigger than prokaryotic cells. Cells generally have three main jobs. So we're going to group all the cell parts into one of these jobs. The first is to make energy. Living things need energy for all activities. They need to clean up the waste produced from making energy. We can't make energy without producing some waste. Cells also have to make protein. So proteins do all of the work in a cell, so we need a lot of proteins. That's a major job. And cells need to make more cells. That is for growth and reproduction, to replace cells that have been damaged. Cells, all living things need to reproduce. So we're going to learn about the organelles that make these three major jobs happen. Organelles do the work of a cell. Here is an animal cell model, and we can see all the different organelles, and we're going to be going through them one by one. Each organelle has a structure or a job to do. These organelles keep the cell alive, and therefore they keep you alive. And as our penguin is saying, they are like mini organs, which is exactly what they are. So from your reading, you should have noticed that there are some organelles which assist with the following: energy and protein. You should take a moment to answer this now. And once you have finished answering, we are going to be answering this just in a few moments as we go through this lecture. So cells need power, and that means they need energy. Energy is going to fuel life, growth. The cell has to basically take in food if it's especially if it's an animal cell. But even plant cells, plants make their own food and they need to take it in as well. They have to digest or break down food. Cells have to take in oxygen. Most eukaryotic cells are aerobic, which means they require oxygen to make energy. And then they need to make ATP. ATP is the molecule of energy. So all cells use this molecule called ATP. We're going to have really a whole chapter about ATP, but for now, you just need to know it's a molecule of energy. So all these things need to happen to make energy. And then, as we mentioned before, we've got to remove the waste. So what do we need to do this job? We need a membrane. We need things called vesicles, lysosomes, and vacuoles. We'll find out what all that is. We need a mitochondria, or several of them. And if you're a plant cell, you will need chloroplasts. So let's begin with a membrane. We already know a little bit about the membrane because we learned about it in our biochem chapter. A membrane separates the inside of a cell from the outside of a cell. So this is going to help a cell control what enters and leaves. So some things are going to have an easy time getting in and out of a cell. Other things are going to have a more difficult time getting in and out of a cell. A cell membrane also needs to recognize signals from other cells. And that's going to allow for communication. Now, something we already learned is that the structure is a double layer of fat, which we already know is called a phospholipid bilayer. If you remember from the last chapter, phospholipids have a polar head. You can see that up here in the upper right. And they have a lipid or a non-polar tail, and those non-polar tails are very long. So the phospholipid bilayer will basically look like this. We have the polar heads on the outside of the cell and the inside of the cell, and the inner part of the membrane is just this vast non-polar area. And that's going to be really important when it comes to this point up here, controls what enters or leaves a cell. Basically, it's going to be the membrane that determines that. Things that are nonpolar are going to have a very easy time getting through the membrane. Oxygen and carbon dioxide are nonpolar things that are going to zip right through that membrane. However, other things that are polar or ionic are going to have a harder time going through the membrane. Like food, most food is going to be polar or ionic. Water is polar, and although it's small, it's still going to have a difficult time making it through that nonpolar inside. Nutrients are usually polar and waste products are polar, and that's going to make things difficult, which we'll talk about more when we spend more time on the membrane. There are also going to be receptor molecules, as you can see here on the outside of the membrane down here in the lower right. These are going to be proteins that receive signals, and that's going to be important as well. So there, outlined in red, is our membrane. Notice it surrounds the entire cell. It's the boundary, it controls what goes in and out, and it recognizes signals. Inside the membrane, that fluid is called the cytoplasm. The cytoplasm is the jelly-like material holding all of the organelles. Now, another group of organelles that we talked about were vesicles, vacuoles, and lysosomes. So these three things are difficult to distinguish, and we will find out more about those. Basically, what are they? They are for moving material around a cell, and they're also for storage. So for example, we have a small food particle here that is being brought into the cell, as you can see, and it's going to be moved around in a vesicle. A vesicle is sort of like a taxi. It moves things around the cell. So it's just a membrane sac. It's just a membrane separating outside from inside. Now here we have a lysosome. This little purple thing here is a lysosome. So a lysosome is a membrane sac that is special because it's filled with digestive enzymes. These little purple dots are supposed to be digestive enzymes. So notice what the lysosome is doing. The lysosome is meeting up with the vesicle. What do you think the digestive enzymes are going to do to the food? Well, they're going to digest the food. Here, a vesicle is meeting up with here a lysosome is meeting up with another vesicle that contains part of an old mitochondria. So again, what do you think the digestive enzymes are going to do to the mitochondria? They're going to digest it, and we're going to end up with digested pieces in this membrane sac. And now we are going to call it a vacuole. Now it's used for storage, so we refer to that as a vacuole. The vacuole is filled with digested nutrients, and that's just stored, and the cell can use those digested nutrients for later. All right. Vacuoles can take different forms in an animal cell. We find a lot of vacuoles that are used for storing food. In certain organisms like this paramecium, some vacuoles are contractile vacuoles. What these do is they have a very special function that takes water in and they squeeze the water out to keep the water balance in the cell at a normal level. And plant cells have one giant vacuole in the middle. You can see it's called a central vacuole, and this is for storing water and other things in that solution. Lysosomes, which are basically just vesicles that contain digestive enzymes, can be used to kill cells when those cells need to be destroyed. Sometimes living things have to get rid of some of their cells. Some cells have to die for proper development in an organism. So here we have a tadpole, and tadpoles have a tail, but at some point the tadpole needs to develop into a mature frog, and that tail has got to go. And so lysosomes are going to help get rid of this tail now that the frog no longer needs it. This process of cell autodestruct is called apoptosis. You see that word highlighted in red. That is the autodestruct process, and it is when lysosomes break open and they basically destroy a cell. The tadpole tail gets destroyed when it turns into a frog. As you can see here, human fingers have webbing early on in fetal development, and that webbing needs to go so that we can use our fingers in a more dexterous way. As you can see here, that lysosomes have caused apoptosis for certain cells so that our fingers do not have webbing anymore. And then, of course, sometimes we see that apoptosis doesn't happen correctly and fingers or toes can be stuck together when they shouldn't be. Okay, so lecture check. Differentiate between lysosome, vacuole, and vesicle. So write your answer now. And when you answer that, you should see that a lysosome is going to be filled with digestive enzymes. A vacuole is going to be used for storing things, and a vesicle is for moving components around a cell. So there, highlighted in red, we have a lysosome. Vacuoles and vesicles are there. Mitochondria are coming up next. So what else do plants need to make energy? They need mitochondria. Mitochondria look like this. It's a double membrane organelle, and that double membrane organelle, the inner membrane is highly convoluted, which gives it a very large surface area. So it kind of looks like almost like a bean with a maze in the middle. Mitochondria take energy from sugar and combine it with oxygen to make ATP. So basically, a mitochondria is where we make all of our cell energy. So all those nutrients that we broke down with the lysosome and using the membrane and the vacuole and the vesicle, that energy, those food particles make their way to the mitochondria along with oxygen. The mitochondria is going to undergo cell respiration, and cell respiration is going to combine the sugar with the oxygen to make ATP. Chloroplasts, on the other hand, are something different that plant cells will have. A chloroplast will look like this. You see it in the corner. I'll move my face out of the way. So a chloroplast is also a double membrane with these little discs on the inside. Those discs are going to be good at absorbing the rays of the sun. Chloroplast is going to make sugar from sunlight, basically. So sunlight is going to hit these chloroplasts, and it's going to combine water and CO2 to make sugar. That process is called photosynthesis, when sunlight and CO2 make a little bit of ATP and, importantly, sugar. And remember, ATP is energy, and sugar is going to be stored energy, so it's like chemical potential energy. All right, that sugar can be used to build leaves. It can be used to build other part plants like roots. Fruit tissue is going to be made out of those sugars. So there are lots of things that can be made. The cell can take those sugars and build cellulose. It can build starch. It can build all kinds of things. So the process of photosynthesis is very important. All right. So lecture check. What processes happen in both the mitochondria and the chloroplast? What kind of organisms have mitochondria? Is it prokaryotes? Is it animals? Is it plants? And what kinds of organisms have chloroplasts? You have the same choices as above: prokaryotes, animals, or plants. Take a moment and answer these questions. All right, so what processes happen in mitochondria and chloroplast? Mitochondria, we have cellular respiration happening, and in chloroplast, we have photosynthesis happening. What kind of organisms have mitochondria? Animals and plants both have mitochondria because they both need to make ATP. Prokaryotes do not. What kinds of organisms have chloroplasts? Only plants. All right, we are going to stop there for now. We'll get to this one in the next lecture, and we'll go on to part 2 of cell organelles.